Good afternoon, everyone. Welcome to the HPL seminar this week. It is my pleasure to introduce our speaker today, Colin Firminger. Colin started his undergraduate in 2007 here at the University of Calgary in mechanical engineering and got bachelor's degree in 2012. And then in 2014, he joined Dr. Brent Edwards' group as a master's student in biomedical engineering. His project was about the metatarsal stress fracture risk in minimalist versus traditional footwear and achieved a master's degree in 2016. Colin has continued his academic career as PhD student under the same supervisor, but now investigating the fatigue behavior of patellar tendon to assess patellar tendinopathy risk. Currently, Colin is planning to complete his PhD course next year and to try to find a job in industry afterward. When I was asking him about the scientific accomplishments that he is most proud of, he mentioned that one of his supervisor experiences that he had worked with an undergraduate student in biomedical engineering, and the student was highly successful in her research and also has written a manuscript that will be submitted soon. Aside from academic background, Colin also mentioned that he's been looking forward to skiing a lot this season. And in addition, he has joined a golf club this year and has done over 40 rounds of golf, but he feels that he still has a long way to go. Today, he will be sharing part of his thesis work entitled as Effect of Cyclic Loading on Mechanical Property Degradation and Fatigue Behavior of Human Patellar Tendon. All yours, Colin.
All right, thanks, Sangwon. So to begin with, we'll just go over what the patellar tendon kind of structure and function is. So the patellar tendon is a fibrous tissue that connects the patella to the tibia. And it's comprised mainly of just longitudinal collagen fibers. So the point of the tendon is to transmit force generated in the quadriceps muscles to the tibia. And as such, it's highly loaded when during activities such as jumping, cutting, and running. So one of the most common injuries that occurs in the patellar tendon is called patellar tendinopathy. This is an overuse injury that has an insidious onset. So athletes with this injury can never identify one acute part of sport or something that caused this injury to develop. It kind of just continuously develops and gets worse over time. And it's most commonly characterized by pain at the inferior patellar pole, so the bottom of the patella where it connects to the patellar tendon. The Blazina scale has been used to kind of define the functional limitations associated with patellar tendinopathy as it progresses, beginning with stage 1, which is just pain only after a sporting activity, advancing to pain that happens at the beginning of the activity that might disappear when the athlete warms up and then might come back when they get fatigued, to, as it progresses, pain pretty much all the time, either at rest or during activity. And this is when it kind of leads to the athlete's inability to participate in sports. And then if you can get this far, it can potentially lead to complete rupture of the patellar tendon. So patellar tendinopathy, there's been a lot of ways that researchers have tried to develop interventions for this. One of the most successful ones has been eccentric loading of the patellar tendon with drop squats. However, one of the main problems athletes face is that symptoms can last more than a year, regardless of what kind of exercise or whatever interventions they're undertaking. So once someone develops patellar tendinopathy, it can be potentially a long road ahead for them to return to sport. So there's been a lot of studies that have looked at epidemiological aspects of tendinopathy, including this one done by Leon et al. In 2005, where they looked at the prevalence of tendinopathy in youth athletes across a range of different sports. And they found that tendinopathy prevalence was highest in sports of volleyball and basketball compared to all these other different sports you can see there. In addition, a study done by Ferretti et al. Looked at the incidence of tendinopathy in volleyball athletes. And they noted that one of the main risk factors they found was the number of weekly training or playing sessions that the athletes were undertaking. So you can see here that as athletes are participating more and more in these either training or playing sessions, their incidence is increasing quite a bit. So if we look at this first set of data, it's kind of telling us that tendinopathy has something to do with the sports that are involving high loading magnitude of the patellar tendon in volleyball and basketball that involve this jumping a lot of the time. And if we look at this second study, it's showing us that potentially there's something there with athletes increasing the number of loading cycles that they're applying to their tendon and the incidence of their tendinopathy that they're experiencing. So the pathophysiology of patellar tendinopathy is it's pretty debated in literature currently, and still to this day, but it definitely starts with repetitive submaximal strains that are being applied to the tendon. So if we don't have these, there's no risk of tendinopathy occurring. These repetitive submaximal strains have been evidenced to lead to microdamage accumulation within the patellar tendon. And this microdamage accumulation potentially has been thought to lead to a biological response that can be observed. And this can take the form of cell proliferation or vascular neuronal ingrowth within the tendon. And at end stages, we see increased proteoglycan production and increased inflammatory markers as well. So whether or not directly from microdamage accumulation or indirectly through this biological response, at the end stages of tendinopathy, when athletes have symptoms, we see this material property degradation within the tendon. And this generally represents itself as reduced tendon stiffness, reduced Young's modulus, and increased cross-sectional area. So if we just look at the pathway of repetitive submaximal strain to microdamage accumulation to material property degradation, we can think of this as mechanical fatigue, where we essentially have some material that's being repetitively strained. We have some sort of damage that's beginning to accumulate in that tissue or material, and that's leading to this degradation of the material properties of that tissue. Now within tendon, it's been shown fairly repeatedly that microdamage is related to this material property degradation. And on the left here, this is a study done by Fung et al. In 2010, where they repetitively loaded rat patellar tendons in vivo. And on this graph here, we can see kind of their methodological overview, where we have the peak cyclic strain that they were applying to the tendon in this green curve here. And they were taking images of the tendon at 3 points. So the low strain time point, the moderate strain point, and then the high strain point. And you can see here that the low and the moderate strains are being taken on this nice linear part of this peak cyclic strain curve, which they call the secondary phase, or the secondary region of this peak cyclic strain curve. And then in the tertiary phase here, as the strains start to increase rapidly before failure, is where they were imaging the tendon for this high strain region. And what they saw, compared to control in the top left corner there, which shows this nice wave-like pattern of the collagen fibers, at the low strain, the top left picture here, we see some kinked fiber deformations. So you can see this kind of ridge running along here where the fibers that run from left to right have kind of been kinked. That's some of the lowest microdamage that they could observe. At the moderate strain time point, they observed worsening of this kinked fiber deformation microdamage, and they also saw this interfiber spacing here beginning to appear between two adjacent fibers. Lastly, at this high strain, they saw complete discontinuities in the collagen matrix. They saw more excessive fiber thinning, and overall just a lot more degradation of the material compared to this control. In another study that was just recently published by Zitney et al., they looked at the percent of denatured collagen over a fatiguing protocol for tendon. And you can see here from 0% of the fatigue life, so the number of cycles to failure, all the way up until 100%, so right before the tendon failed, they captured this percentage of collagen denatured. And you can see here that there's this nice increasing relationship between how long the tendon was under load or how many cycles were applied and the amount of collagen denaturation. But even more interestingly, they showed that when creep strains, so the amount of plastic deformation of that tendon was plotted against the amount of denatured collagen that they observed within the tendon, there's this region basically up until about 4% of creep strain where there's not a ton of denatured collagen going around. But then after that, there's almost this linear increase in the amount of denatured collagen that we can observe. So it's pretty much directly related between the amount of creep strain or plastic deformation that's occurring in the tendon and the percentage of denatured collagen that's being observed. So the effects of fatigue on tendon material or mechanical properties have been well established with different tendons, including this study done by Ren et al. On a human Achilles tendon. And the first thing that's generally observed is a decreased Young's modulus over time. So if we look at this graph, we have strain on the x-axis of the tendon, stress on the y-axis, and each one of these loops is just one cycle of loading, and they repetitively loaded this tendon until it failed. Each of these loops represents a different percent of the fatigue life, so 1% all the way up to 99%. And we can see here that the Young's modulus, which is just basically the slope of the linear part of the curve near the top, we can see that it's fairly stable between 1% and 50%, and then it starts to decrease at 90% and even more at 99% of the fatigue life there. The second thing that's commonly observed is increased cyclic energy dissipation. So cyclic energy dissipation is basically just the hysteresis of each one of these cyclic loops, the difference between the loading and the unloading sides of the curve. We can see here that, again, it's fairly stable as we go from 1 to 50%. And then at 90%, it starts to increase. And then at 99%, it increases even more. And the final thing that's commonly observed is increases in peak cyclic strain. So just like what we were looking at with the Fung study, as peak cyclic strain was increasing, we can see here as well, this plastic deformation starts to increase. So from 1 to 10%, it increases a little bit. From 10 to 50%, it increases a bigger magnitude. And then from 50 to 90% and 90 to 99%, it increases quite substantially. So there's large increases to peak cyclic strain right before the tendon is about to fail. So based on this mechanical property changes that exist from fatigue, different studies have tried to quantify predictors of fatigue life. This one on the left was data that's from Sheckman and Botter, who fatigue loaded extensor digitorum longus human tendons until failure at a bunch of different stress magnitudes. And they plotted the stress magnitude on the x-axis versus the fatigue life that they measured on the y-axis. And they come up with this nice relationship here between the two. You can see here that as stress magnitude increases, the fatigue life decreases. And it's important to note that this is a highly nonlinear relationship, similar to what Dr. Miller was talking about last week, because the y-axis here is a logarithmic axis. Back to the Renadal study on human Achilles tendon, they were looking at initial nominal strain versus fatigue life. And they also found that a nice nonlinear relationship existed between the amount of initial nominal strain that was applied on the tendon and how long it could last until it failed in fatigue. Two measures that haven't been explored for tendon, but might be potentially interesting to look at are peak cyclic strain rate and stiffness degradation. So on the left here, we have this traditional looking peak strain versus number of cycles curve that I've shown you a few times already, where we have this nice linear region in the middle, and the slope of this region is the peak cyclic strain rate. And this might be a potentially good metric for predicting the fatigue life of tendon, as well as stiffness degradation, which is defined as basically the degradation of Young's modulus over the number of cycles. So the Young's modulus of the current cycle divided by the initial Young's modulus measured, and it comes up with a graph that looks something like this, or we can take the slope of that graph in the middle point where it's most linear, and this potentially could be a good prediction as well for fatigue life. It's interesting to note that tendon fatigue behavior is or has been shown to be site-specific. So this work done by Kerr et al. On different wallaby tendons from different parts of the wallaby body, it shows this. So in the leftmost graph here, the researchers took a bunch of different tendons from different parts of the body and estimated the stress in life that these different tendons face, starting from lower tendons or lower loaded tendons like the tail and the toe extensors, all the way up to highly loaded tendons like the gastrocnemius and plantaris tendons. And what they did was they loaded these tendons in fatigue and measured at a test stress of 50 megapascals firstly, and measured the time that these tendons took to rupture. And you can see here that the tendons that had low stresses in life failed fairly quickly. However, the tendons that had high stresses in life actually lasted a lot longer until they ruptured. And this is for a given test stress of 50 megapascals, regardless of whatever the tendon stress in life was. However, when the researchers tested each tendon at its estimated stress in life from that leftmost graph there, they found that there was almost no relationship anymore, and all the tendons failed at a similar time to rupture, regardless of where they were located in the body. So this kind of indicates that the tendons, at least in the wallaby, had been adapted to the stresses they faced in daily life, and their fatigue behavior is kind of evident by that. So a recent methodological advance in the quantification of strain in mechanical testing is with digital image correlation. And this has been applied to tendon in a few different times. basically this metric consists of spray painting the tendon with a stochastic speckle pattern that you can see in the left images on either side, and tracking that speckle pattern as the tendon and the material is loaded. And basically once that speckle pattern is tracked, we can estimate the deformation at every point on that surface, and then we can from that estimate the strains across the surface. So we can see here that for the Achilles tendon and also for the porcine patellar tendon, there's this highly heterogeneous strain distribution across the surface that appears. So we thought that this might be interesting to pair with a fatigue test, as it might show the changes in this heterogeneous strain distribution throughout the fatigue test as the tendon is fatiguing. So with all that being said, the purpose of this work was to quantify the mechanical fatigue behavior of the patellar tendon using both nominal and digital image correlation measures. We hypothesized that in similar to the other studies that we've looked at, Young's modulus would decrease and energy dissipation would increase as the fatigue loading progressed. And we also hypothesized that measures of stress, strain, creep rate, and damage rate would all be highly correlated with fatigue life via power law or nonlinear relationships. So this is the setup that we used for the cyclic loading. We had 19 cadaveric patellar tendon samples that were excised from cadaveric knees. And for these samples, we kept the patellar tendon attached to both the patella and the tibia. So it was attached to both bones. And what we did was we flipped it upside down so that the tibia was now on the upper portion and the patella was in the lower portion, and inserted it into this clamping setup here. So we machined down the tibial tuberosity, or the tibial diaphysis, so that could be held in the clamps, and it would leave the tibial tuberosity where the actual patellar tendon inserts into the tibia outside of the clamping region, so we wouldn't have to worry about any effects of clamping directly to the tendon. And we did a similar thing with the patella at the bottom. We clamped the patella into a clamp that could be rotated, and we rotated it to 146 degrees, which represented the knee angle, or the patella angle at peak strain during a counter movement jumping exercise that we recorded in some previous research. So this is the setup here. I'll show you a quick video of what the actual fatigue loading looked like. So basically it's in force control. So it's loading between 2 megapascals and a specified peak stress over and over again. And we're hydrating that tendon with a few different saline drips just to keep it moist. You can see the speckle pattern on it too. And it's important to note also that we remove the medial and lateral sections of the tendon just to decrease the cross-sectional area, so that the stresses that we applied to it would result in a lower force that we needed to exert on the tendon, as some of the pilot studies that we were examining or using actually started failing in the bone before the tendon. So we decided to reduce the cross-sectional area there so that we needed to apply less force to get to a certain stress. So this was the cyclic loading and digital image correlation protocol. And basically the graph we're looking at here is just a representative sample. The number of cycles to failure on the x-axis and the stiffness in newtons per millimeter are on the y-axis. So initially what we did was we took a pre-fatigue digital image correlation acquisition. So we loaded the tendon to whatever maximum or peak stress we were going to apply. And we took a video of the speckle pattern as it was being loaded with our DIC camera. And following this, we immediately started the cyclic loading protocol. So the protocol is shown here. It's between a minimum 2 megapascals and a maximum, which ranged between 7 and 36 megapascals, which represented between 11 and 60% of the ultimate tensile strength of the tendon. And we just basically watched the stiffness degradation here. So Kmax represents the maximum stiffness observed for that tendon, and it starts to degrade at this point. So at 90% of that maximum stiffness, we paused the test and took another DIC acquisition. So we took another video up until that maximum stress, and then we continued it right after, allowed it to keep degrading until 80% of the maximum stiffness, where we took a third DIC measurement, and then allowed it to degrade another 10% to 70% of the maximum stiffness, where we took our final DIC measurement or video. And then after that, we allowed the tendon to keep going until it outright failed, which is where the X is there. So the outcome measures from this cyclic testing were, first of all, to subjectively take a look at the changes to these surface strain patterns that we got from the digital image correlation videos. Next, to look at changes to Young's modulus over the fatigue life of the tendon, and we defined Young's modulus here as the slope of the stress strain curve between 50 and 100% of the loading. We also looked at the cyclic energy dissipation throughout the fatigue life, which again we defined as the difference between the area of the loading and unloading curves, so basically the hysteresis there. And then we also looked at the relationship between fatigue life and several measures. So the first one was peak stress, which again was between 11 and 60% of the ultimate tensile strength. And then we just calculated that as the applied force divided by the area. We measured the area at the mid-substance of the tendon with digital calipers. The next measure was initial peak nominal strain. So to get this measure, we took a virtual extensometer from our digital image correlation images, and we put it in post-processing software that came with the digital image correlation. So this virtual extensometer basically just spanned the entire length of the tendon there, and that was our measure of nominal strain initially, so before the fatigue testing occurred. We also grabbed measures of initial median strain and initial 99th percentile strain, and this was from the surface strain distribution map. So you can see here an example of one of the surface strain distribution maps that we get from the digital image correlation. And if we look at this as a histogram of different strain values and the number of pixels, we could just take the median value for that from the initial DIC measurement and plot that against fatigue life. And we also took the 99th percentile over here of that histogram and plotted that against fatigue life too. And finally, we took the creep rate and plotted it against fatigue life. Again, that's just the linear portion, the slope of the linear portion of that peak strain versus cycles curve. And we also took the slope of the damage curve, which we call the damage rate. So initially, to look at the changes that occurred in the digital image correlation, we'll start with the pre-fatigue image of a representative sample that we see here. So blue represents areas of low strain, and red or yellow represents areas of increased strain. And this is the pre-fatigue measurement, so the digital image correlation before any cyclic testing had happened. And then as we go to the right, we're increasing in the amount of fatigue that's been applied to the tendon. So 90%, 80%, and 70% of that maximum stiffness value. So we can see here that one of the main takeaways is that obviously the strains tend to increase regardless of magnitude. They're kind of across the entire surface of that digital image correlation map. We see an increase in strain as the tendon fatigues more and more. But more importantly, what we're seeing is that certain areas in the tendon, such as this location here, we're seeing an increase in not only the magnitude of the strain concentration, but it's also increasing in size as the tendon fatigues further and further. And we pretty much saw this across all samples. It wasn't always in the same location here, but we tended to see an area or areas of increased strain develop, and they would also, throughout the fatigue test, they would increase in magnitude and size. In terms of the Young's modulus throughout the fatigue test, we found similar things to Renadol, where this Young's modulus significantly decreased. So this plot here is showing again that strain and stress on the y-axis. So this is just the normal stress strain curves, starting from 0% of the fatigue life all the way up to 100% of the fatigue life. And again, we can see subjectively that the Young's modulus tends to stay fairly constant up until about 60 to 70%, where then it starts to slightly decrease up until 100%, where it's decreased quite a bit. If we look at the average data across all of our samples, percentage of fatigue life is shown on the x-axis, Young's modulus is shown on the y-axis here. We see an initial significant increase in Young's modulus from 0% to 10%, followed by a fairly consistent decrease here, with significant decreases starting at 70 to 80, 80 to 90, and 90 to 100%, where there's a sharp decrease right before failure. We think this initial increase in fatigue or in Young's modulus is mainly due to the tendon just seating itself within the clamps. And this has been observed in other studies as well. In terms of the cyclic energy dissipation, it also showed a significant decrease throughout the fatigue life here. If we look at the overall average data again, over percentage of fatigue life versus the cyclic energy dissipation on the y-axis, we can see that similar thing where it decreases initially between 0 and 10%. However, it was a little bit different from the Young's modulus in that it stayed fairly consistent throughout most of the fatigue life. And then right near the end, between 80 to 90%, and then 90 to 100%, it started to decrease fairly rapidly. So this is a little bit in the contrast of the Young's modulus, which tended to decrease a little bit more throughout the most of the fatigue test, whereas this kind of stayed more constant. So in terms of the predictive variables for fatigue life, we first looked at peak stress, and it displayed a very good relationship with fatigue life. So we're seeing here fatigue life on the y-axis in a logarithmic form, and then peak stress on the x-axis. And we do have this highly nonlinear relationship that develops with an R-squared of 0.65. So it's explaining 65% of the variance there. In terms of initial peak nominal strain, so again, that was the strain that we measured with the virtual extensometer as kind of our nominal measure. Against fatigue life, it did a little bit worse than peak stress, but it still was a good measure with R-squared of 0.57. Yeah, and then compared that to the initial median strain here, which again was just the median strain of that digital image correlation strain map we saw. We almost observed the exact same relationship between initial median strain and initial nominal strain. So the equations are almost identical to each other. However, initial median strain displayed slightly more variance compared to initial nominal strain. It's interesting to note that initial 99th percentile strain, so that 99th percentile of the strain distribution, had a very poor relationship with fatigue life with an R-squared of only 0.07. So it was not a very good predictor of the fatigue life of the tendon. So in terms of creep and damage rates, they were actually both significantly correlated with fatigue life. And they both had very, very strong relationships. So you can see here the creep rate, which again is that slope of the curve, the peak strain curve. It had an R-squared of 0.96. So it was pretty much almost perfectly correlated with the fatigue life of the patellar tendon. Whereas damage rate also had a very good relationship with an R-squared of 0.86. So slightly worse than the creep rate, but still one of the strongest relationships that we saw with all the variables that we examined. So initially I told everyone that we expected the fatigue behavior of the patellar tendon to be different from different tendons in the body because of the different stresses in life that they experience. And it's interesting to know what happens when we plot these two, the results from these two different tendons together. So first here we have the data that we just collected in this study of the patellar tendon here versus initial peak strain, peak nominal strain value. And if we add the data from the Achilles tendon here, we can see that it looks like it was tested in almost a different regime of fatigue loading. So the data from the Achilles tendon was tested between 1 cycle and about 10,000 cycles at its peak, representing the low to moderate fatigue regime. Whereas the data that we collected was between about 10,000 cycles and almost 1 million cycles. So it was almost in the high loading cycle regime. But if you look at these side by side, it almost looks like they could be part of the same kind of behavior essentially. And if we take one curve and plot them together, we actually are able to, it looks like it fits the data pretty well, and it gets an R-squared of 0.78, which is greater than either the two single alone. And it's interesting also to note that the slope of the Achilles tendon versus the slope of the patellar tendon, when statistically analyzed, they're actually not significantly different, although the numbers might look a little different. So potentially the differences that we're seeing between these two tendons is not due to the actual fatigue behavior. It's more just due to the regimes of fatigue loading that they were tested in. So we found in this study that strain and strain rate were both fairly good estimators. Strain rate was a very good estimator of fatigue life, but also strain was a good estimator of fatigue life. And why would this be the case? And we mainly think that this is because microdamage is predominantly strain controlled. So in tendon, microdamage manifests as fibrosliding initially, and then once that begins to fail, the molecules, the actual collagen molecules, begin to slide against one another. And once that mechanism begins to fail, the collagen molecules actually start to unfold. And this is all as strains increase. Additionally, once this happens, whole fibers that reach a certain strain start to fail, and these transfer load via crosslinks to the remaining fibers. So essentially, this is the entire mechanism of failure in tendon seems to be highly strain controlled, and this makes sense with what we're seeing. Additionally, it's been pretty consistently shown that tendon failure occurs between nominal strains ranging from 13 to 20%. So this again gives us fairly good confidence that once the tendon reaches a certain strain, nominal strain value, it basically just, it cannot maintain that structure and it begins to fail. One of the main limitations with this study was that we didn't account, or there was no adaptation repair that could be accounted for during fatigue testing, as we were testing the tendon outside of the body. It has been shown in tendon that following exercise, there's increased collagen synthesis biomarkers. And following a lengthy exercise protocol of multiple weeks, we see the tendon actually hypertrophy with increased cross-sectional area and increased stiffness as well. So obviously this cannot be accounted for in our relationship, but it may influence the actual fatigue behavior of the tendon. So if we look at the fatigue relationship that we obtained between the patellar tendon here, and then also adding in the Achilles tendon to get this overall relationship between initial peak nominal strain and fatigue life here, I just did a quick back of the hand calculation and estimated the strain during running at about 4.4%. And using that equation between strain and fatigue life, that gives us a fatigue life for the patellar tendon of about 3,400 cycles. So if there's no repair adaptation going on, that would be about a 10-kilometer run until the tendon fails outright. So obviously that's not happening in reality. However, we believe this behavior is still relevant for sure with regards to the mechanical fatigue behavior of the patellar tendon. So the take-home messages from this study were that the fatigue life was very well explained by nominal strain and also the creep rate. And also that the Achilles and patellar tendons, although their stresses in life are different, they appear to have similar fatigue behavior. And this could be because they're both very highly loaded during locomotion and different activities. Future directions for this research would be to non-invasively track continuous changes potentially in tendon, as this may prove very effective for preventing overuse injuries, as this is highly related to the fatigue life of the patellar tendon. With that, I'd like to thank my lab group and supervisory committee, and then the sponsorship.
Thank you very much, Colin, for the great talk. Before I open the seminar to discussion, as it was announced last week, I would like to kindly encourage you to turn on your web camera if you feel comfortable with it. I believe it would allow us to have more active atmosphere of interaction during discussion period, I think. I much appreciate you doing it. So if you have any question to Colin, please use the raise hand button that you can find it if you go to click the icon labeled participants at the bottom center of your screen. You will see the blue icon raise hand button at the bottom on the right side. The floor is now open for questions. Yes, Ross, please go ahead.
Hey, Colin, this is Ross Miller. Nice job. It was a great talk.
Thanks.
I was super interested in what you just said at the end about how the data suggests that failure of the tendon would occur during a 10-kilometer run. And acknowledging that these were like 75-year-olds who maybe don't have the strongest tendons in the world. And you also trimmed away, some of the tendon there. But does that suggest that the healing, like the time course of healing, must be pretty fast, like it's occurring literally during the run?
Again, I think a big part of the, like what you said, is that these were older tendons and they're definitely like weaker than what we would see in vivo. But there has been some evidence that at least repair is happening on the order of days afterwards. We're seeing increased upregulation of certain pro-collagen markers within 1 day of a fatiguing protocol. So I think it's happening fairly quickly after exercise. Repair is happening quickly, and then adaptation has been shown up to 72 hours and 7 days following as well. So there's definitely repair and adaptation occurring. And as with all mechanical testing of biological tissue, I think we are underestimating the fatigue life a little bit just by the nature of in vitro testing.
Thanks.
Okay, John, please go ahead.
My question was a lot like Ross's. I was thinking about that early slide that you had that with the volleyball players working out 4 or more times a week. The issue would be the balance between damage accumulation and healing, right? And so if you really understood how long it took to heal, you could probably work out what the best training rate would be. but then it would probably depend a little on just how vigorous the workout was, right?
Right. Yeah, exactly. It's definitely like a balance between the two. And it's the regenerative and healing ability of tendon is still kind of, it's not well known, I think, right now. So I think that's an important next step for sure.
Thanks.
Okay, Eric, please go ahead.
Hi, Colin. Nice talk today.
Thanks.
So I was wondering, you are testing actually a ligament and a tendon in both of these scenarios. So do you think that, that your results expand to other ligaments within the body in terms of their failure rate?
Are you saying ligament because it's attached to two bones? Is that what you mean?
Mm-hmm.
Yeah, I think it's more relevant to tendon because the patellar tendon it has more of the behavior of a tendon, even though it's attached to two bones. It originates in development directly from the quadriceps, and then the patella kind of comes in after to act as like a sesamoid bone, or to increase the mechanical advantage essentially of the tendon. So in that sense, there's studies that have shown that it behaves and its biological makeup is more of a tendon. So I don't think ligament is completely different, and I think that this is somewhat relevant for ligament, but I think it's more for tendon for sure.
Yeah. Thanks.
Okay, Siska, do you have a question for Colin?
Hi, yeah, thank you. That was really interesting. I really enjoyed it. I have two questions. The first one is, as you were saying before, just about the persons, like the females, if you have any more information about what they did, or if you just have their age and anything like that. And then I was wondering, because you're saying that you saw that the strain was different depending on the location, so you saw different strains, and how you would explain why that would be, where you saw more strain and where there was less, and what that means.
Yeah, totally. So in terms of the donors, we had basic information about height, weight, and stuff like that, but not about their activity level. So I can't really speculate on that too much. In terms of the strain distribution, I think we just assume a lot of the time that tendon is this homogeneous material, that it just experiences the same amount of strain everywhere. But I think at least what these findings are showing us is that there's different levels of tissue quality even within a given tendon, and certain regions will fail sooner than others based on different lengths of fascicles that might strain more than others initially and stuff like that, and different regional areas of tissue quality that are different from one another.
And sorry, just where did you see the most strain? Was it, which part was it? Because
there was never like there was never a consistent region where strain was higher in one over the other. It would kind of tendon to tendon, it was kind of specific. So there's usually some sort of concentrations near the entheses, and then within the mid-substance of the tendon, it was kind of varied.
Okay, thank you.
Okay, Dave Hart, please go ahead.
Enjoyed your talk, Colin. In vivo, and is there a preferred location for failure? Rupture?
You know what? I'm not exactly sure about that. I mean, the pain is fairly consistent in tendinopathy in the same location at the, like, right where it inserts into the patella, but I'm not sure about actual outright rupture. Yeah, I'm not sure.
In your sample set from the older females, did they fail at one particular location, or was it an avulsion, or did it tear, or
yeah,
the insertion?
So for both females and male samples that I tested, we saw it generally looked to me like the tendons were pulling out of both entheses. You could hear popping sounds. And then when the tendon actually separated fully, you could see a lot of the tendon was still attached to the patella side and a lot was still attached to the tibia side, but the pieces that were attached were close to the entire length of the tendon. So it looked like it was almost pulling out from the bone at one end or the other. It wasn't like failing right in the middle or anything like that.
Okay, so I guess that's a transition point because the antices have a composition that's somewhat different than the
Right.
the major part of the tendon. So it's kind of like where tears occur in the supraspinatus, et cetera. It's right near the insertion. So how is that related to the failure properties of the bulk of the tendon versus the weakness or the vulnerability of the antices?
That's a good question. We couldn't measure the properties of the antices, right? So I'm not sure exactly what's happening there, but yeah, I can't really speculate about exactly what's happening at the antices other than that the strains, generally there were strain concentrations near there. So higher strains would likely mean that the tendon was likely to fail in that location. It was interesting to note that we were almost concerned about it failing because the bone was too weak at the antices region. So we actually did a small little post hoc study looking at quality of the bone and then trying to match that to the fatigue life, and we saw no relationship there. So it's definitely related to the tendon strain more so than anything else, but exactly what's happening at the antices, I'm not sure.
Okay. Thank you. Well, give it some thought. The question may come up again.
Okay.
Okay, Philip, please go ahead.
Regarding, let's go back to, let's say, volleyball and jumping. So what is the influence, or let's say, or what is more relevant concerning the fatigue life? Is it more the takeoff phase or the landing aspect of a jump?
Well, I'm trying to think back to the study that Olivia and I did at the beginning of my PhD. I think we were seeing the highest strains during it all comes back to strain essentially, right? It's where the strains are the highest. And I think we were seeing the highest strains during landing, I'm pretty sure. They were around, I think it was an average of like 4.4%, but strains were still high during both takeoff and landing. But I'm pretty sure they're a little higher during the landing. But regardless, it's still about at the tissue level, whatever you're doing at the macroscopic scale with your body, it's what the effect that has on the strains at the tissue level that's really influencing the fatigue life there.
Okay, thanks.
You're welcome.
Yeah, I saw Benno, you raised your hand earlier. You have a question?
Yes, Colin, thanks for the presentation.
Welcome.
If I understood correctly, you constructed the measuring device so that you can rotate the patella. Why did you do that?
Just to give it a more physiologically relevant loading scenario. So during jumping, which is one of the activities that's been highly linked, or athletes who jump a lot are linked to patellar tendinopathy. We thought it would be a good idea to mimic what the tendon is experiencing during a jump, essentially, at the highest, where it's being loaded in the highest strain during a jump. So using that data from the study I was just talking about with Olivia for the NBA, we just found the angle of the patella to the patellar tendon at that time point where the strain was the highest, and that's what we chose to go with.
So
and that's gonna, sorry?
Your load was asymmetric then?
It was, I guess in a sense you could say it was asymmetric from anterior to posterior, because the anterior fibers would have been potentially stretched more than the posterior fibers because the patella was rotated a bit. And studies have shown that there's a strain concentration that develops, like if the patella is like this and the anterior side's here and the posterior side's here, there's a strain concentration that kind of develops in the middle there. And it can get worse with flexion of the knee, essentially. So yeah.
So did you leave the patella free that he could adjust, or did you fix it at a certain angle?
Yeah, we fixed it at a certain angle at 146 degrees, which was the angle that corresponded to the time point during a counter movement jump where the strains were highest on the patellar tendon.
Okay.
Yeah.
Okay, Brent, please go ahead.
More of a, I guess, a comment than a question. And it's sort of playing off Ross's comment, which we've had discussions all week about this idea that, you know, how quickly does a tendon have to adapt, and nobody gets tendinopathy after, or tendon rupture after, you know, a 10K or whatever. But I also was thinking about it, and you know, one thing that came to mind is, you know, I don't know how you come to this conclusion that the strain is 4% or whatever during running.
Yeah.
But if that's just sort of back of the envelope calculation, if you're off by 10%, then you're off by the number of cycles to failure by 100%.
Yeah, that's a good
So
point.
if
I mean,
it's not 4% and it's 3.6%,
yeah,
you're not failing after a 10K, all of a sudden it's a 20K. And so
yeah,
when you start thinking about the margin of error that you have with, you know, this sort of exponential type, this power law curve, you know, it's easy to sort of say, oh, well, but if I'm off by this much, it has this huge influence. So
right. And our ability to actually estimate strain during running, who knows how accurate that actually is, right? So.
Right. And then the other thing is like, you say 4%, I feel like I remember that paper, and I feel like the strains were like 2 to 3%.
That's during.
And then I'm trying to figure out, but you said running. So are the strains at the patella higher during running than they are during drop landing?
I doubt it. I just used some data from that winter study, 1990, that showed the force in the patellar tendon that they estimated using some musculoskeletal modeling, and then I converted that to a strain based on some stiffness measures that we got from ultrasound. So it wasn't.
So there's going to be error in that calculation too, right?
There's a lot there, yeah. So yeah. I guess for us to say exactly at 10K, it's going to fail. That's very hand wavy and not specific. So yeah.
Yeah. Okay. All right. Thanks.
Mm-hmm.
Art, please go ahead.
Yeah, I have some questions about the cycles,
Okay.
and I guess I'm gathering that the cycles are not all the same throughout the experiment because you're showing the stiffness is changing and stuff like that. Mm-hmm. So, okay, so I'll just start with one question, which is the work done in each cycle changing, or how does it change as you're fatiguing the tendon?
I guess you're exerting the force over a larger distance because it's in force control. So
well, I mean the network in the sense of you presumably have a hysteresis
right
in the work loop.
Right. I mean the hysteresis it's increasing right over time.
Uh-huh.
Not necessarily. I mean, you have the data. You showed the plot. Bring it up.
Right. I guess it's fairly constant for a lot of the tests, and then it becomes increases.
You called it energy dissipation, I think, Colin.
Yeah.
Okay. I guess. Well, I see. So it's just in the graph there.
Yeah. So this is the average of all the specimens that we tested there. So
okay, I guess I would say it doesn't seem to change very much until the very end.
Yeah.
So I'm just curious what is going on in tendon, and this kind of reveals how little I understand what's going on. Mm-hmm. And I guess the question is, first of all, they say that in normal human behavior, you'll stretch your tendons and then you'll get something like 90% energy return. But my question is, let's say this is at fairly low strain, so we don't think there's any damage going on or very little damage. Mm-hmm. So if you're losing 10% of your energy, what is happening there? Like, how is, I'm presuming that is, if you're doing work on the tendon, then that has to show up as heat. So that means you have to have a mechanism to convert that into heat, or you need to have that strain energy, change the structure, right? So then you're doing work,
Right.
to change how the fibers are aligned or something. So what's going on? This in sub-fatiguing cycles, what's happening?
I mean, there's definitely some evidence that there is irreversible sliding of the fibers and fibrils. So at multiple length scales, that's happening. So that you have fibrils that are connected to each other with crosslinks, and then within even the molecules, those molecules are connected to each other with crosslinks. So that seems to be, there's evidence that that's the initial mechanism of the tendon basically taking on fatigue damage. So these start to slide against each other, and those crosslinks start to break and lengthen. And I think that's where some of that might be coming from. And then after that, there's actual stretching of the molecules themselves. It's irreversible. So the molecules they're in this like triple helix. So it looks like a little DNA of 3 collagen molecules that wrap around each other. That'll actually unravel. And it's a progressive thing, right? So there's different lengths of fibrils. So as the tendon increases length and more fibrils will actually be engaged, and some will get to a certain length that they fail, and then they'll no longer contribute to the load sharing, and that'll be passed on to other fibers and stuff like that. So I think it occurs across multiple length scales, and there's multiple different mechanisms that's going on there.
And when you say sliding, you mean when that happens that causes crosslinks to break, or do you mean that sliding also has some friction between, I don't know, 2 fibers are sliding against each other? And
I think it could be, I'm not sure about the friction, but I think that probably might occur. But there's definitely crosslinks stretching and then breaking between fibrils. Yeah.
So then even at fairly low strains where we don't think we're fatiguing the tendon, you would say that there are crosslinks being broken. And so basically, even at that level, some damage is occurring.
Yeah, it's interesting that it's been shown a few times that at low levels of fatigue, we do see some microdamage that accrues in the tendon, but this doesn't come out as changes to the actual mechanical behavior until much later on, when the tendon's got a lot of microdamage accumulated within it. So yeah, that's occurring for sure.
Okay, well, that makes me wonder about rubber, and just ask, how does tendon compare to rubber? Because rubber, you can make a tire and then you can drive 200,000 K. I mean, there are tires that will last a long time. And then what's different about that kind of rubber versus tendon?
I think tendon can repair itself and rubber can't. But I'm
talking about rubber that lasts for a really long time.
Right. I mean, I think there's always this balance of, movement and strain on a biological material, whether it's tendon or bone, causes some sort of microdamage to accumulate. And if that microdamage rate of accumulation is less than the ability of the tissue to repair itself, then you wouldn't even notice that anything's happening, right? It's only when that rate of damage is greater than the rate of repair that things might start to break down potentially.
Yeah, but I'm actually curious why. So rubber in a tire is not being repaired, and it still lasts a long time. And is that because it's just at much smaller strains, or what's the difference there?
I'm not sure about the tire. I can't really speculate on it, but I mean, a lot of the time with tendon, if it's below at least a nominal view of the tendon, if you strain it to less than 4% strain, it's said that, or around 4% strain, it's said that you don't really see any reduction in modulus or anything like that. It's only once you start to strain it after do you see like actual plastic deformations and stuff. So it could be perhaps that the tendons are adapted to kind of stay below this threshold. I'm not really sure about the rubber though. I'm not sure why there'd be that huge difference, but.
I see. And then I'll just ask one last thing, which is, does collagen have rubber-like elasticity?
In what sense?
Well, rubber-like elasticity is stuff where when you stretch it, it actually cools down. I guess the example that people give is you take a rubber band and you can press it against your lips, and then you can stretch it, and then your lips are pretty sensitive to temperature, so you can tell the temperature change. And it has to do with the mechanism of elasticity, which is that so in rubber you have these fibers that are kind of in these random orientations, and then so if you stretch some rubber, and then if you let it sort of pull back on you, what's actually happening is that it's just Brownian motion, is that you have heat that is just striking these strands, and then that heat in all sorts of random ways tends to get the rubber strands to be more bunched. And so rubber is very different from like metal, where you're pulling on the bonds, between the grains of the metal. And so the question is that how collagen works? Is it also something where heat is the mechanism to make it sort of recoil?
I am not sure about that for tendon. I would speculate that it behaves less like that, more like a traditional, like a more engineering material, but I am not sure.
Okay, well, I'm just curious about all these things because to me it sounds crazy that running a 10K would be anywhere near putting you into the failure range. And then what Brent said about, yeah, you could be 100% off. I still feel like that seems very far off. Like I think running a 10K, I think my guess is that you're quite far off from failure. Mm-hmm. And then and I'm just thinking, what's happening, because if you have 90% energy return, 10% is still quite a lot of energy. And then if you're doing 10,000 cycles, each of them with a 10% energy loss, that seems like there's a ton of strain energy there. And then I want to know how much of that is, ending up as heat with no damage versus how much of it is causing damage.
Yeah, it's interesting. I haven't really thought about it from that point of view before. But it does appear that there's quite a bit of energy dissipation within the tendon. And I think that's been shown at the Achilles tendon too. It was originally thought to be this like really good spring, but I think it's more being shown to be a method of keeping the muscle at a more isometric state than actually acting to return a ton of energy. But, yeah.
Okay, thanks.
You're welcome.
And Quan, please go ahead.
Hey, Colin.
Hey.
Thanks for the great presentation. It's very clear. I really enjoyed it.
Thanks.
I have a question regarding your data. The one that you show in one of your last slides. Can you show it?
Yeah, sure. What are you looking for?
This one.
This guy?
Yeah. So it appears that your data is has less spread compared to the data in the black dots. So what is the strategy that you use to decrease that spread? Because I think your data is cleaner than the others.
Thanks for that. I don't think it's actually, I think it's just it's an illusion of the logarithmic scale here. So Brent and I were talking about this too, but like this is pretty much one order, or maybe the range of this is from like 10-ish to 100,000, and ours is going from like 100,000 to 1 million. So it's just, I think it's purely the logarithmic scale. The only other thing I would potentially say is that in this Ren study, they were clamping directly to the Achilles tendon because it's like Eric pointed out, it's only attached to the calcaneus and the other end is attached to the gastrocnemius muscles, right? So they had to actually clamp onto it, which they and many others have shown that it kind of presents a lot of challenges there. And I think they had some failures kind of near that part, whereas we got to clamp right to the bone. So I think that it reduced our scatter a little bit, but overall it was pretty similar.
Yeah. Okay. Great. So the second question I have is regarding to the loading axis. I presume you set up in such a way that the loading is applied uniaxially.
Yeah.
Along the axis of the tendon.
Yep.
But for patellar tendon, normally the loading axis is not coaxial for both ends, right? Depending on what flexions you have for your knee.
Right. So that's kind of why we chose to rotate the patellar tendon a little bit to kind of make it a little more physiologically relevant. And that would change the strains from like the anterior side to the posterior side a little bit.
Yeah.
But that's pretty much like the best we could do, we think, for that. Yeah, we weren't changing the angle throughout the test or anything like that.
So my question is, if you were to load the same sample, which we don't have this hypothetical situation,
yeah,
a little bit off the axis of rather than having the uniaxial, but you switch it a little bit sideward, make it not coaxial. Do you think the fatigue life will change due to that?
Potentially, yeah, it could. If you're loading one side more than the other, then that side might fail sooner because it's being kind of isolated potentially. And then that might just kind of increase the failure rate of the tendon basically by causing higher strains to be transferred faster to the other remaining fascicles. But I think anterior to posterior, I think it's a little better. Maybe if you didn't align it properly, like if you had the patella like kind of tilted one way so that laterally was being pulled more than medially or something like that, maybe that would affect it. I'm not sure, but I don't think that happens really physiologically. So.
Okay, thank you.
Mm-hmm.
So Colin, I also got a question from Tim, but he doesn't have a microphone, so let me just read it for you.
Okay.
Does DIC on the surface of the tendon accurately reflect the strain deeper in the tendon?
That's a good question. I think potentially, but not a guarantee for sure. So some studies that have done finite element modeling of tendon have shown that the strains actually, they change throughout as you go deeper into the tendon. And that's basically because the tendon is not being loaded perfectly equally at every point, right? What Dr. Hart was talking about with the anthesis and stuff, it causes some parts of the tendon to be loaded more than others given a certain angle of knee flexion or flexion of the tendon. So best case scenario, they're close, but likely they change throughout as you go deeper into the tendon.
Thank you. Walter, please go ahead.
Thanks, Colin. I enjoyed your talk very much as well. And having run many 10Ks and worried about it as well, same questions everybody else had. And I guess a couple of things that I was thinking about is how the loading was applied that you give. You know, I think I'm running, I'm loading my patellar tendon for 200 milliseconds, and then I have a flight phase, and then I have the other leg, and then I have another flight phase. So I'm getting, I don't know, 600, 700 millisecond rest. Yep. So how was actually your loading done?
So it was continuous, right? So it was a sine wave at 2 hertz, basically.
Yeah, okay.
So 2 times per second was being loaded.
So do you think then if you loaded more like a run is, so that you load for 200 milliseconds and then you give it, I don't know, 600 millisecond rest and then you load again, do you think that would affect the cycle to failure numbers compared to what you're doing?
Yeah, potentially. So the fatigue life of tendon from other studies like this Ren study, they've compared it with just creep data. So where the tendon is just held at a certain force under, or it's held under a given force until failure, basically. So there's no rest at all. And they've shown that the fatigue failure, or the failure of tendon in cyclic loading, is both a function of the cyclic loading and also the time under load. So less time under load would potentially, or spreading that time out under load would potentially extend the fatigue life. And just subjectively speaking, when I paused the test to take the DIC measurements, the tendon did recover a little bit during that time when it wasn't being loaded. It was like a 5-minute, stop of the fatigue test just to take the one-off DIC measurement, and it recovered a little bit. So potentially.
The other thing I was thinking about fatigue life, many years ago when I first came to Calgary, Dr. Cy Frank and Nigel Shrive, they were doing lots of ligament testing. And one thing they realized is that when they used, later on then they developed this humidity chamber. So they had this 100% humidity that surrounded the ligament, but it was not water, but it was actually fully saturated 100% humid air. And that seemed to make an enormous difference to a variety of the mechanical testing that they did. And so how was your environment?
Yeah, so ours was definitely not submerged in water, but we tried our best to keep it hydrated because that is definitely a concern. So I had 4 saline drips kind of dripping on the tendon the entire time just to keep it moist throughout the entire test, basically. But yeah, other studies have actually submerged the tendon completely in like saline or something solution to actually keep it hydrated.
Yeah, because I always thought that was a brilliant idea to have this 100% humidity chamber. Just something maybe to think about.
Yeah,
the other thing that I thought was fascinating is when you showed that graph where you said that's about the expected loading or strain that tendons have normally, and then they all ended up being between about 10,000 to 100,000 cycles to failure, if I remember correctly. Do you think that actually tendons might be built like that, that they're built to last for a magic number, somewhere between, let's say, 100,000 cycles, and if you load it differently, it adapts in a way that number stays constant? Do you think there's some kind of a magic there about predicting?
I think it could be adapting in a way that it's decreasing. So if you're applying more load to it and the strain increases, it adapts in a way that increases the stiffness, right? So that a given force lowers the amount of strain that the tendon sees, and that's commonly observed as an adaptation. So I think it's probably trying to stay in this like nice strain regime where it can last for a extensive period of time. And then if you start loading it out of that, or if you start loading it under that, it'll adapt accordingly, I think.
And another question, well, really more a comment, was regarding Art's idea, about the heat. And you probably know that people have made heat measurements. I'm particularly familiar in horse racing, where supposedly in certain limb tendons of the horse, temperature goes up quite dramatically, to the extent where some people say it might be getting so high that there might potentially some collagen degradation because of the heat, that might contribute to the thing. Yeah.
Okay.
So I assume you didn't measure heat since you didn't answer anything, but I thought but it seems that in vivo, when horses run a lot, or presumably people as well, there is indeed some heat accumulation that's substantial, and that can be measured. Yeah.
Okay.
And just a last thing, because I know we have done similar things, and one of the things I always find difficult is to get a really good cross-sectional area. Can you just quickly tell me how you got cross-sectional area?
Yeah, it was a fairly basic measure. We just took, tendon is normally fairly rectangular shape, but because we cut off the medial and lateral sides, it was actually very rectangular shape. So we just took 3 measurements of width and 3 measurements of thickness with just digital calipers, and then assumed a cross section or a rectangular cross section and got it that way.
So then you calculated it, so you didn't use some kind of a caliper thing or so, try to measure it actually or so?
No, I mean, other studies have done either like using ultrasound in vivo to estimate it, or MRI or something like that. And potentially we have MRIs of this data, so we could potentially use that, but because we cut off the medial lateral sides, I guess it wouldn't really be a great measure. So yeah. Stress wasn't really like, so that was basically used to calculate a stress, right? But that wasn't
exactly
our main outcome. Our main outcome was the strain that was developed by that stress. So we weren't as concerned with it, but it's a valid point for sure.
But if I remember, on some occasion, you applied a certain amount of stress, and of course, if your cross-sectional area was,
right,
10% or 20% off, then you might think you applied the same stress, whereas you might have
right,
applied 10% or more or less than what you actually think.
Yeah, that's true. It's definitely a limitation for sure.
Yeah, very nice. Thanks, Colin.
Thank you.
Dave, would you have any question for Colin?
Well, I had a couple of questions, but I also had a comment for Art that that the sliding that you talked about appears to be influenced by the expression of PRG4 on the edges of the fascicles, and PRG4 is the molecule that's also produced by the superficial zone of the cartilage. So it's probably contributing to the ability to slide. And it turns out in tendons, PRG4 expression decreases as you age. Interestingly, in some rabbit models that we looked at, we found the opposite occurs in the medial collateral ligament. As they get older, the PRG4 increased, but still can't explain that. The second thing is, in the real world, in some studies we did with Karim Khan and Alex Scott long ago, there's additional cells in the tendon, particularly in the peritoneum or epitendon or whatever you want to call it.
Yep.
So there's some mast cells and other cells that appear to increase as the integrity of the collagen matrix becomes less in asymptomatic patellar tendons, humans. So in the real world, there are some potential other factors at play that could contribute to the integrity of the matrix. And the last point I guess I would make, Colin, is that there is some discussion that the problem of tendinopathy, say in patellar tendinopathy, is that it's actually an injury because of a lack of fidelity of the jumping and landing movement. In your model, it's the fidelity is controlled, so that actually it's not necessarily related to the fatigue properties, but it actually is related to the more often you exercise it or participate in a practice, say in volleyball, the more opportunity there could be for an inappropriate landing to start to affect the enthesis.
Okay. Yeah, I hadn't thought about it in that kind of way, but I think that could make sense too if there's just like an insulting factor that starts this kind of cascade, whether it's biological or not. But yeah, okay, cool.
Okay, Kun, do you have any question for him?
I guess so. Hi, Colin. I was intrigued by this curve that you were looking at with Art. I don't remember the title of the slide. Oh, it was about this energy dissipation, right? So
yeah,
I guess that part of that energy is going into shifting these curves progressively to the right, right?
Yeah, so like the plastic strain.
So clearly there is some, even though this energy dissipation remains constant for a large part of the fatigue life, I guess that part of that energy is going into changing the, well, what Art said, like changing the shape or changing the configuration of these fibers such that this whole curve shifts to the right.
Right.
Which I think is just amazing, because if I look at the strain amplitude in one cycle and I compare that to the amount that the entire curve shifts to the right, that's just fascinating to me. So I was wondering, do you think this actually happens in real life in our tendons?
Like, are they lengthening as we use them more, or as we overuse them?
Well, just if you run it, I mean, I'm not sure to what extent the stress that you present here is comparable to like the stress during running, but I imagine that at the end of a 10K, if this data is what it is, then at the end of a 10K, my
floppy tendons
strain is hugely larger than at the beginning, which is just fascinating to me. Yeah.
Yeah, I mean, the data suggests that your tendon there will be some plastic deformation through cyclic loading, right? So I'm not sure to what extent it's going to happen in your body. That's highly dependent on the stress or strain magnitude that you're applying to it, but there should be some extent of lengthening, I guess, over time if you're consistently doing this over and over again. Yeah.
Okay, and just one more question about this, just to make sure that this is not a weird experimental artifact. So this is the measurement that you're showing here. Is this based on your machine, or is this based on some measurement of the tendon?
This is nominal, so this is coming from the machine's displacement versus
so it is
force.
possible that actually the bone is shifting a tiny little bit in the gripper.
It could be, yeah, it could be for sure.
That
I think
is
between
causing
0
this
and 10%,
creep,
that's
right.
Between 0 and 10%, I think that's a large reason for why it shifts so much.
I see.
And then after that, there definitely could be some additional movement or slippage of the bones. But I think it's pretty minimal because they seeded pretty well within those clamps after they've been going for a while. So
okay, sure.
Yeah.
Okay. So this is real what we're looking at.
Yeah,
that's
for sure.
Amazing. Yeah. Yeah. Okay. And so this leads me to another point, which is just a fun thing, that as a Dutchman, I am traumatized by Dutch soccer players taking penalties in world championships of soccer. And one of my theories about why we are so bad at it and why people in general are bad at taking penalties at the end of a match is that you are super fatigued at the end of the match, and then apparently your tendon is in a totally different state. And then you rest for 15 minutes, you stroll to the penalty shooting spot. So you have no idea what the state of your body is, and then you take a penalty and you hopelessly miss. So
yeah,
I think we should advocate as a group for a new protocol in penalty shooting where you do a 5-minute warm-up before you shoot the penalty.
I like it.
Okay.
I don't think that's the only problem that the Dutch have.
Okay, I'm willing to concede that point, Benno, but still.
Okay, Rob, do you have any final question for Colin?
Yeah, it's probably a very similar question to Dave Hart had. At the beginning of your talk, Colin, you said that if athletes were training 4 times a week instead of once a week, they had an increased rate of this problem with ruptures. And if you could put up your slide that you had before with the Achilles tendon and the Patellar tendon. That one, yeah. I'm wondering if there's an alternate hypothesis, and therefore why you looked at 10,000 or 100,000 cycles. That if you train more times a week, is there an alternate hypothesis that your muscles are simply, it's muscular fatigue that's put you in the position, I think as David was saying, that you're perhaps more likely to make an error and more likely to be on an initial peak strain for what you've got here at about 0.09 or 0.1, which means you only need to cycle, 5 or 6 of those and you've got a rupture. And whether, 2 questions, whether that hypothesis is at least as likely as having to go to 100,000 cycles. And then separately, how hard is it to stuff up when you land and actually produce a peak strain of 0.09 or 0.1? Is it actually difficult to be that bad? And if you land awkwardly, is it that your patellar tendon has a cross section like this, but the strain, because you've landed poorly, it's just in this little piece that it is in fact 0.1? So I guess there's 2 questions.
Yeah, so I think, first of all, to get a peak strain of like 10%, that's a lot of force you have to apply to the tendon. So like we were doing maximal counter movement jumps for athletes and we were only getting around like 4%. So I think even if you screw up that pretty bad, I don't think you're even close to getting to 10%. I think Ren was applying forces that high, but that doesn't mean they're physiological, right?
Right.
So I think that's that kind of situation. In terms of like insults from like one high loading cycle, I mean, in reality, we're never just going to consistently load the tendon at exactly 4% strain, right? It's always going to be different from jump to jump and stuff like that. And there's different ways of handling that, different kind of failure methodologies of taking like the probability of failure at a given strain, and then how many times you go for that strain, and the probability of failure at the next strain you jump at, and how long you can last at that strain, and kind of summing up those different probabilities to get an overall estimate of what's going on. I haven't thought about it from the way of like, like one high load starts the damage that then progresses to tendinopathy. I think we don't know enough about the pathophysiology to really know if that's the case or not, but I think there's strong evidence that there's definitely like mechanical fatigue as a pathway of injury. So that's just kind of what we've chose to do here, but the other way could definitely be a possibility.
And someone, if I can steal another question, in isolation, you're doing 10,000 or 100,000 cycles.
Yep.
But without any repair cycle, without sitting in a nice solution and at a nice temperature. How difficult would it be to get an experimental animal to do this? Get a rat to, I mean, mice, I think, travel 7 kilometers a night on a wheel, but how difficult would it be to put train rats to work incredibly difficult, train more than 4 times a week, to train, 6 days a week with extremely high loads, and get to what you think is 20,000 cycles, and then take half of them and get half of them and look for damage, leave some for a week and go, okay, this has got to be repaired. They should be the same as the first batch, but there's been a week's worth of repair. Has anybody put an experimental animal like a rat into that sort of circumstance?
Dave has, Dr. Hart. I think with the rat supraspinatus downhill running stuff, right? I think you guys did that. And it was like a model of tendinopathy, making rats run downhill for an extended period of time for a while, and then inducing basically tendinopathy from that kind of a system.
Actually, with Walter and Joanne Archambault, we tried to do that for the Achilles using what's called a Backman model, which was developed by some people at Umeå in Sweden. But it hasn't been used for some time now over here. I think that was about 20 years ago. But it is possible to induce changes to the Achilles tendon using that kind of passive use of the Achilles tendon. Do you remember, Walter?
Yes, I remember very well, yeah. And I remember it was very hard, very hard to really get any damage done. And that's why we then actually slightly changed the protocol. But another comment that I wanted to make to Colin, you know, suggesting the downhill running. So we just recently did that, didn't look at tendon, but looked at muscles and joints. With having mice doing a lot of downhill running, and we found damage. But the problem was we also found a tremendous amount of systemic inflammation in these animals. And it seems when you have them run downhill a lot, you cause a lot of damage in the muscles that then somehow spews out some myokines that puts in a whole bunch of inflammatory markers that Dave knows much better than I into the system. And so then these downhill running mice, they almost looked like our fat rats, metabolically speaking. And then the question becomes how much of the damage that we see was actually due to the loading of the downhill running, and how much was due to the fact that these mice that ran downhill compared to uphill or level were metabolically very different. Lots of inflammation.
Yeah, and inflammation's been kind of targeted with tendon as well. At least at the initial stages of tendinopathy, there's this inflammatory response that tends to subside as it gets to more degenerative states of disrepair. So it's definitely like an interplay for sure. It's not just mechanical, it's not just biological. I think it's definitely a bit of both for sure.
Okay, Colin, I think this is, we are getting slowly wrapping up since it's close to 4:30. So I would like to thank Colin again for sharing your work. Next week, we'll be hearing from Dr. Paris Vaikil, and the title of her talk is Correlations between changes in stress on the surface of the articular cartilage and the development of focal damage signaling, the onset of post-traumatic osteoarthritis in an ovine model. Thank you for coming today, and I'll see you all next week.